Once you start, you really know your model, and once you have really, let's say, perfected it, the opportunities for analysis are endless. You can do a lot of different experiments. You can do cytokine measurements, which is also something I plan to do, oxygen measurements, TM measurements, live cell imaging, infection progression. You can do endless experiments with it. So all you need is patience and practice. Hello everyone, so I am Simranpreet Kaur. I am a doctoral researcher in Jena at the Friedrich Schiller University and Leibniz-HKI, and I work on organ-on-chip models to study invasive lung infections, especially with mucormycosis. So it's the mucorralian fungi, namely Leishmania, mucor, and rhizopus. So we know that traditionally disease modeling is either in 2D cell cultures or maybe in animal models. But both have their own cons. So for 2D cell cultures, the morphology of the cells is not actually accurately depicted, whereas in mice models or animal models, they have their own ethical problems associated with it. So that's why we wanted to have a model that is an interface between both. So it's a step up from the 2D culture, but not yet an animal culture. That's why we decided to do organ-on-chip model. And also the other thing is that I decided to choose this fungus because mucorralian fungi are not very well researched. There is not much research into it and the infection associated with it. So we really wanted to see how the host and pathogen interaction is occurring during this kind of infection, how the host is reacting and how the pathogen and the fungi, how they are behaving. My model is based on studying invasive mucormycosis, which is a severe pulmonary infection. So the whole idea behind the model is to do live cell imaging of the infection progression so that we can clearly see the lung epithelium cells, the blood endothelial cells, how the fungi are behaving and how the macrophages are behaving. So to establish the model, we use the chip from Dynamic 42. It is the BC002 chip, which has two chambers separated by a porous membrane. So in one of the chambers we seed our HUVECs, which are our blood endothelial cells, and then after a few days we seed A549. So these are the cell lines we use for lung epithelial cells, and then we have the very preliminary model established for us. To these we have our own donors, healthy donors, from whom we draw blood and isolate the PVMCs, which are then differentiated into macrophages and then added to the lung compartment to make up a teeny tiny alveoli on chip model. To this model, then we have a perfusion setup, so to have media perfusing through the vascular side, which mimics blood flow, and eventually we infect the model with the fungal spores, where it is 1.5 hours of static, you can say static infection, and then 3.5 hours of infection via perfusion. And then eventually these chips are then stained and imaged depending upon if we want to do live cell imaging, we want to do endpoint staining, and that's how we analyze our chip. So currently for the organ-on-chip model, since I am establishing it for a particular fungi, it takes a lot of optimization. So now we have finally arrived at what cell lines we are going to use, which will be the A549 cell line, which is a lung adenocarcinoma cell epithelial cell line for the lung cells, epithelial cells. And then for the blood side, we are using the HUVECs, which is the standard cell line everybody uses, and these are our controls. And we saw that after 5 hours of infection, the Leishmania, mucor, and rhizopus, for both Leishmania and mucor, there was already germination within 5 hours. And a really surprising event that we see is that these hyphae, when they are germinating from the spores, when they come in close contact to the porous or the pores of the membrane, they are branching and then causing angioinvasion into the vascular side. So we saw these both for Leishmania and mucor, and as you can see here, they are also germinating out of a pore in the endothelial compartment. Whereas for rhizopus as well, we saw this, but I didn't see much germination on the lung side. However, they have been phagocytosed by macrophages. So already within 5 hours of this infection, the macrophages are phagocytosing the spores, the fungi is doing its infection. And then the next step would be for us to actually quantify this, which we are in the process of currently, to calculate hyphal lengths to see how much angioinvasion occurs. And this will give us more insight into what infection progression is like. Yeah, so I think organ-on-chip models, they might look very daunting in the beginning because they have so many components involved. There are so many different cell lines, so many different immune cells, so many different pathogens that you have to introduce, and there are so many factors that you could go wrong. But I think the only thing you need is patience. Patience and practice. The more you practice, the more easier it gets. And I think once you start, you really know your model, and once you have really, let's say, perfected it, the opportunities for analysis are endless. You can do a lot of different experiments. You can do cytokine measurements, which is also something I plan to do, oxygen measurements, TM measurements, live cell imaging, infection progression. You can do endless experiments with it. So all you need is patience and practice.